Gene amplification and polymerase chain reaction. Gene amplification, also known as gene duplication or chromosomal duplication, is a cellular process in which multiple copies of genes are produced. The result is an amplification of the phenotype or expressed as trait associated with the gene. Phenotype is used to describe observable and measurable characteristics of an organism. In a simple sense, it may be duplication of a region of DNA that contains a gene. Gene duplication occurs because of a serious genetic flaw in a cell or a group of cells. Gene amplification has a serious implications to evolutionary history as well as to drug resistance in cancer cells and major concern in biology for many other reasons. Most cases of gene amplification occur during an error in the homologous recombination when two similar strands of DNA exchange genetic information. This occurs during many genetic processes but is most common in meiosis. Gene amplification or a duplication is believed to play a vital role in evolution. Susumu Ohno was the most famous developers of gene amplification in his book Evolution by Gene Duplication. Ohno argued that gene duplication is the most important evolutionary force since the emergence of the universal common ancestor. It is believed that the entire yeast genome underwent duplication about 100 millions of years ago. Plants tend to undergo gene duplication or an amplification more commonly than animals. For instance, wheat is hexaploid, a kind of polyploid, which means that it has 6 copies of its genome. When genes are amplified and traits are strengthened, the strengthened traits are inherited by offspring, that is, organism produced by one or more parents, and passed on to subsequent generations. This is a very significant consideration in evolution. Significance in evolution. Not all gene amplification has great importance to evolution. Sometimes the amplified trait dies with a single organism that experiences the gene duplication. This happens when a gene is overexpressed and the trait it codes for is expressed to level that is unhealthy to the organism. It can also happen when the duplication occurs in a somatic cell rather than in a sex cell. Somatic cells are cells that make up of an organism except for sexually reproductive cells. The genetic information in somatic cells are not passed on to offspring. Thus, it does not present itself in later generations. Impact of gene amplification in other areas. One of the most widely examined aspects of a gene amplification is its role in drug resistance in some diseases. Cancer cells, for instance, often express considerable drug resistance because of the amplification of the gene that prevents the cancerous cells from absorbing the chemotherapeutic drugs completely. Particularly, amplification occurs in a gene that codes for a protein that is able to selectively pump materials out of the cancerous cells. This protein has tendency to pump chemotherapeutic agents, successfully neutralizing the treatment in many diseases. Detection of gene duplication. Technologies such as genomic microarray. Microarray is a 2D array on a glass slide, also known as genomic hybridization, are used to detect chromosomal abnormalities such as microduplications in a high throughput fashion from genomic DNA samples. In particular, DNA microarray technology can simultaneously monitor the expression level of thousands of genes across many treatments or experimental conditions, greatly assisting the evolutionary studies of the gene regulation after gene duplication. Coming to the PCR. PCR is nothing but polymerase chain reaction. Polymerase chain reaction, better known as PCR, is one of the technologies that not only made a tremendous impact on the scientific community but also affected many aspects of our everyday lives. More than 30 years ago, the introduction of recombinant DNA technology as a tool for the biological sciences revolutionized the study of life. Molecular cloning allowed the study of individual genes of living organisms. However, this technique was dependent on obtaining a relatively large quantity of pure DNA. This depends on the replication of DNA of plasmids or other vectors during cell division of microorganisms. Researchers found it extremely laborious and difficult to obtain a specific DNA in a quantity from the mass of genes present in a biological sample. A technique that amplifies the DNA through a simple enzymatic reaction was developed by Carey Mullis at that time, which enabled scientists to make millions or even billions of copies of DNA molecule in a very short time. Thus, polymerase chain reaction reaped the highest scientific honor for its inventor in record time, as it proved a solution to one of the most pressing problems facing biology at that point of time, the replication of DNA. In 1993, Mullis got Nobel Prize in Chemistry for his dedicated work on PCR. It is the polymerase enzyme that drives the PCR. A polymerase will synthesize a complementary sequence of bases to any single strand of DNA, providing it has a double-stranded starting point. This is very useful because you can choose which gene you wish the polymerase to amplify in a mixed DNA sample by adding small pieces of DNA complementary to your gene of interest. These small pieces of DNA are known as primers because they prime the DNA sample ready for the polymerase to bind and begin copying the gene of interest. During a PCR, changes in temperature are used to control the activity of the polymerase and the binding of primers. To begin the reaction, the temperature is raised to 95 degrees centigrade. At this temperature, all double-stranded DNA is melted into single strands. The temperature is then lowered to 50 degrees centigrade. This allows the primer to bind to your gene of interest. Thus, the polymerase has somewhere to bind and can begin copying the DNA strand. The optimal temperature for the polymerase to operate is 72 degrees centigrade. So, at this point, the temperature is sometimes raised to 72 degrees centigrade to allow the enzyme to work faster. There are now twice as many copies of your gene of interest as when you started. The cycle of changing temperature 95 degrees centigrade, 50 degrees centigrade and 72 degrees centigrade is then repeated and 2 copies become 4. Another cycle and 4 becomes 8. Up to 30 to 35 cycles after amplifying your gene into millions of copies, it is possible to run the amplified DNA out on the agarose gel and stain it with a dye to visualize it. The bigger the visible band contains more copies of gene of interest that you have created. Coming to the working principles of PCR. As the name implies, it is a chain reaction. A small fragment of DNA section of an interest needs to be identified, which serves as a template for producing the primers that initiate the reaction. One DNA molecule is used to produce 2 copies, then 4, then 8 and so forth. This continuous doubling is accomplished by specific protein known as polymerase. Enzymes that are able to string together individual double DNA binding blocks to form longer molecular strands. To do their job, polymerases require a supply of DNA building blocks, that is nucleotides consisting of 4 bases, that is adenine, thymine, cytosine and guanine. They also need a small fragment of DNA known as the primer, to which they attach and building blocks as well as longer the DNA molecule to serve as a template for constructing the new strand. If these 3 ingredients are supplied, the enzymes will construct exact copies of the templates. The another step is denaturation. Double-stranded DNA templates denature at temperature that is determined in part by their GC content. The higher the proportion of GC, the higher the temperature required to separate their strands of the template DNA. The longer the DNA molecules, the greater the time required at the chosen denaturation temperature to separate the 2 strands completely. If the temperature for denaturation is too low, or if the time is too short, at 80 rich regions of the template, DNA will be denatured. When the temperature is reduced later in the PCR cycle, the template DNA will re-anneal into fully native condition. In PCR, catalyzed by Taq polymerase denaturation is carried out between 94 to 95 degrees centigrade, which is the highest temperature that the enzyme can endure for 30 or more cycles without sustaining excessive damage. In the first cycle of PCR, denaturation is sometimes carried out for 5 minutes to increase the probability that long molecules of template DNA are fully denatured. However, this extended period of denaturation temperature is unnecessary for linear DNA molecule, as it may be deleterious sometimes. Denaturation for 45 seconds between 94 to 95 degrees centigrade is routinely used to amplify linear DNA molecule whose GC content is less than 55%. The another step is annealing of primers to the template DNA, or annealing step. The temperature used for the annealing step is critical. If the annealing temperature is too high, the oligonucleotide primer anneal poorly, if at all, to the template, and the yield of the amplified DNA is very low. If the annealing temperature is too low, non-specific annealing of primers may occur, resulting in the amplification of unwanted segments of DNA. Annealing is usually carried out 3 to 5 degrees centigrade, lower than the calculated melting temperature at which the oligonucleotide primer dissociate from their template. The another step is polymerization for template DNA. Extension of oligonucleotide primers is carried out at or near the optimal temperature for DNA synthesis, catalyzed by the thermostable polymerase, which is the case of Taq polymerase, works around 72 to 78 degrees centigrade. In the first 2 cycle, extension from one primer proceeds beyond the sequence complementary to the binding site of the other primer. In the next cycle, the first molecule are produced whose length is equal to the segment of the DNA delimited by binding sites of the primers. From the third cycle onwards, this segment of DNA is amplified geometrically, whereas longer amplification products accumulate arithmetically. The polymerization rate of Taq polymerase is 2000 nucleotides per minute at the optimal temperature, that is 72 to 78 degrees centigrade, and as a rule of thumb, extension is carried out for a 1 minute for every 1000 base pairs of the product. For the last cycle of PCR, many investigators use as extension time, that is 3 times longer than the previous cycles, apparently to allow completion of all amplified products. Number of cycles. The number of cycles required for the amplification depends on the number of copies of template DNA present at the beginning of the reaction and the efficiency of the primer extension and amplification. Once established in the geometric phase, the reaction proceeds until one of the components becomes limiting. At this point, the yield of the specific amplification products should be maximal, whereas non-specific amplification products should be barely detectable, if at all. This is generally the case after 30 cycles. In the PCRs containing 105 copies of the target sequence and Taq polymerase, efficiency falls 0.7. At least 25 cycles are required to achieve acceptable levels of amplification of a single copy target sequences in a mammalian DNA templates. Parameters that affect PCR. Essential components of PCR. A thermostable DNA polymerase to catalyze template dependent synthesis of DNA. Depending on the ability, fidelity, efficiency to synthesize large DNA products, a wide choice of enzyme is now available for routine PCRs. Taq polymerase remains the enzyme of choice. 0.5 to 2.5 units per standard 25 to 50 microliter reaction. The specific activity of most commercial preparations of Taq polymerase is 80,000 units per milligram of protein. A pair of synthetic oligonucleotide to prime DNA synthesis. Design of oligonucleotide primer being the most important factor that influences the efficiency and specificity of the amplification reaction. Careful designing of the primers is required to obtain the desired product in a high yield to suppress amplification of unwanted sequences and to facilitate subsequent manipulation of the amplified product. The primer should not be too long or too short, which allow inappropriate binding. Deoxynucleoside triphosphates DNTPs. Standard PCRs contain equimolar amount of all 4 DNTPs. Concentration of 200 to 250 micromolar of each DNTP recommended for Taq polymerase in reactions containing 1.5 millimolar magnesium chloride. In a 50 microliter reaction, these amounts should allow synthesis of 6 to 6.5 microgram DNA, which should be sufficient even for multiplex reaction in which 8 or more primer pairs are used at the same time. High concentration of DNTPs, that is more than 4 millimolar, are inhibitory, perhaps because of the sequestering of magnesium ions. However, a satisfactory amount of amplified product can be produced with the DNTP concentrations as low as 0.5 to 1 picomolar to 20 micromolar of an amplified fragment of 1 kb in length. To avoid problems, stocks of DNTPs should be stored at 20 degrees centigrade in small aliquots that should be discarded after the second cycle of freezing or thawing. During long term storage at minus 20 degrees centigrade, small amounts of water evaporate and then freeze on the walls of the vial. To minimize changes in the concentration, vials containing DNTP solution should be centrifuged after thawing for a few seconds in a microcentrifuge. Divalent cations. All thermostable DNA polymerases require free divalent cations, usually magnesium, for activity. Some polymerases will also work but less efficiently with the buffers containing manganese. Calcium ions are ineffective because DNTPs and oligonucleotides bind magnesium. The molar concentration of the cation must exceed the molar concentration of phosphate groups contributed by DNTPs and primers. It is therefore impossible to recommend a concentration of magnesium that is optional in all circumstances. Although a concentration of 1.5 millimolar of magnesium is routinely used, increasing the concentration of a magnesium to 4.5 millimolar to 6 millimolar has been reported to decrease non-specific priming in some cases and to increase it in others. The optimal concentration of a magnesium ion must be determined empirically for each combination of primers and template. The preparations of template DNA should not contain significant amount of chelating agents such as EDTA or negatively charged ions like phosphate, which can sequester magnesium ions. Buffer to maintain pH. Tris chloride adjusted to pH between 8.3 to 8.8 at room temperature is included in standard PCRs at a concentration of 10 millimolar. When incubated at 72 degrees centigrade extension phase of PCR, the pH of the reaction mixture drops by 7.2 pH. Monovalent cations. Standard PCR buffer contains 50 millimolar KCl and works well for amplification of segments of DNA greater than 500 base pairs in length. Raising the KCl concentration to 70 to 100 millimolar often improves the yield of short DNA segments. Template DNA. Template DNA containing target sequences can be added to the PCR in single or double stranded form. Closed circular DNA templates are amplified slightly less efficiently than linear DNA. Although the size of the template DNA is not critical, amplification of sequences embedded in high molecular weight DNA, that is more than 10 kb kilobase pairs, can be improved by designing the template with a restriction enzyme that does not cleave within the target sequence. The importance of PCR. PCR is rapid, inexpensive and simple way of copying specific DNA fragments from minute quantities of source DNA material, even when that source DNA is relatively poor quality. It does not necessarily require the use of radioisotopes or toxic chemicals. There are two reasons why you may want to amplify the DNA. One is, you may want to simply create a multiple copies of a portion of a DNA, which is very rare. For example, forensic scientists may need to amplify a small fragment of DNA from a crime scene. The second one, you may wish to compare two different samples of DNA to know which is the more abundant. Because the DNA is microscopic, so you cannot see which sample contains the most DNA. However, if you amplify both the samples at the same rate, you can calculate which sample was the biggest in the beginning. The third one is, more exotic PCR techniques are used for different purposes. For example, PCR using mRNA template employs reverse transcriptase PCR, that is also popularly called as RT-PCR, to see the mRNA quantity. Therefore, real-time PCR or a quantitative PCR almost replaces microarray technology for mRNA expression profile. Nested PCR overcomes a few problems associated with a large number of a PCR cycle to error-prone synthesis. Inverse PCR is employed wherever the desired target sequences lies outside this region by employing restriction fragment plus flanking sequences essentially inverted. RAPD PCR is useful in genomic fingerprinting. Thus, the PCR technology are many and having diverse application. Thank you.